Biology Exam Preparation for Medicine Students Part 2. In prokaryotic cells, what is the function of the sigma factor?
The correct answer is initiation of transcription. In prokaryotic cells, the sigma factor is a subunit of RNA polymerase that is critical for recognizing specific promoter sequences on the DNA. It ensures the RNA polymerase binds to the correct start site, initiating transcription accurately. The sigma factor does not participate in DNA replication, RNA capping, which does not occur in prokaryotes or protein folding. Once transcription begins, the sigma factor typically dissociates from the RNA polymerase to allow elongation of the RNA strand.
What is the function of telomerase in eukaryotic cells?
The correct answer is extending telomeres. Telomerase is an enzyme that adds repetitive nucleotide sequences to the ends of chromosomes, known as telomeres. This process counteracts the natural shortening of chromosomes that occurs during DNA replication, particularly in cells that divide frequently, like stem cells and germ cells. It does not repair DNA damage, unwind DNA, or synthesize mRNA. Telomerase is crucial for maintaining chromosomal stability and preventing premature cellular aging or senescence.
In the lac operon of E. coli, what occurs when lactose is present but glucose is absent?
The correct answer is operon is fully activated. When lactose is present, it binds to the repressor protein, preventing it from blocking the operator. Additionally, in the absence of glucose, cyclic AMP, SAMP, levels increase, allowing the SAPCAMP complex to bind to the promoter and enhance transcription. As a result, the lac operon is fully activated, leading to the production of enzymes necessary to metabolize lactose. If glucose were present, the operon would remain repressed due to catabolite repression, prioritizing glucose metabolism.
Which phase of the cell cycle is characterized by DNA synthesis?
The correct answer is S phase. During the S phase, synthesis phase, of the cell cycle, DNA replication occurs, resulting in the duplication of the genetic material to ensure each daughter cell receives an identical copy of the genome. The G1 phase involves cell growth and preparation for DNA synthesis. The G2 phase focuses on preparing the cell for mitosis, while the M phase is when mitosis and cytokinesis occur. The S phase is essential for maintaining genomic integrity during cell division.
In plants, what is the main function of the Casparian strip in root endodermal cells?
The correct answer is prevents backflow of water. The Casparian strip is a band of suberin and lignin found in the cell walls of root endodermal cells. It acts as a barrier, forcing water and dissolved nutrients to pass through the plasma membranes of endodermal cells rather than flowing back into the soil through the apoplast pathway. This selective barrier ensures that essential nutrients are absorbed efficiently and transported to the vascular tissues. It does not facilitate osmosis, assist in nutrient storage, or enhance photosynthesis directly.
Which type of genetic mutation is least likely to affect the final protein product?
The correct answer is silent mutation. Silent mutations change a nucleotide in the DNA sequence, but do not alter the amino acid sequence of the protein due to the redundancy of the genetic code. As a result, the final protein remains unchanged in structure and function. In contrast, nonsense mutations introduce a premature stop codon, truncating the protein. Missense mutations alter a single amino acid, potentially affecting protein function. Frameshift mutations result from insertions or deletions that shift the reading frame, often causing severe disruptions in the protein product.
Which protein is primarily responsible for clathrin-coated vesicle formation during endocytosis?
The correct answer is dynamin. Dynamin is a GT posse that pinches off clathrin-coated vesicles from the plasma membrane during endocytosis. It forms a helical structure around the neck of the budding vesicle and uses GTP hydrolysis to sever the connection. Clathrin provides the structural scaffold for vesicle formation, but it is not directly responsible for severing the vesicle. Actin and myosin are involved in cytoskeletal functions, while tubulin is a component of microtubules. Dynamin is essential for completing the vesicle formation process.
Which organelle is responsible for the beta oxidation of fatty acids?
The correct answer is mitochondria. Beta oxidation, the process of breaking down fatty acids into acetyl-CoA units, occurs in the mitochondrial matrix. These acetyl-CoA molecules then enter the citric acid cycle to generate ATP. Lysosomes are involved in macromolecule degradation, ribosomes synthesize proteins, and chloroplasts are responsible for photosynthesis. The mitochondria's role in energy metabolism makes it the powerhouse of the cell.
Which of the following statements about the genetic code is true?
The correct answer is it is redundant. The genetic code is described as redundant because multiple codons can specify the same amino acid. For example, the codons UU and UUC both code for phenylalanine. The genetic code is not overlapping, it is universal across most species, and it contains stop codons, UAA, UG, UGA, that signal the termination of translation. This redundancy helps minimize the effects of certain mutations on protein synthesis.
Which molecule acts as the terminal electron acceptor in aerobic respiration?
The correct answer is oxygen. During aerobic respiration, oxygen serves as the final electron acceptor in the electron transport chain. At complex 4, electrons are transferred to oxygen, which combines with protons to form water. NAD+ and FAD are electron carriers, not terminal acceptors, and ATP is the energy currency of the cell, not involved in electron acceptance. Oxygen's role is vital for maintaining the flow of electrons and driving ADP production through oxidative phosphorylation.
Thank you for listening. If our podcast has been valuable to you, we would greatly appreciate it if you could leave us a 5-star rating and write a review on Spotify and Apple Podcasts, or subscribe, like, and share on our YouTube channel. Your support helps us greatly with discoverability. We hope to see you soon.